Good morning, everyone. I am welcome you all to this 21st day of CSIR online training. Today we are going to discuss about the remaining part of plant physiology. Before entering the session, please let me know, am I audible? Can you hear my voice? And it's visible, the slides are visible. Please type it in chat box. Am I audible? Yeah, thank you for your response. We will start the section. Sorry for the delay. I met some technical problem in OBS, so only I'm late. Okay. Today, remaining part of the unit 6 is the photo respiration, how it is sense for light and air. The leaves and the whole plant will act depends on the light and water uptake, all of the things. So now in this section we are going to discuss about the photosensor, the plant sensory activity, how it is sense the light, how it sense the water, that kind of things. Then another part is phytochemical synthesis. These plants are producing these secondary metabolites. So that secondary metabolites are called as the phytochemicals. In that, how it is that are produced in the plant, that things also we are going to discuss in the today class. Then another one is stress. What kind of stresses are there in a plant? Okay, photomorphogenesis. This photomorphogenesis means these plants acting the or moving the leaves, and plant movements are depends on their light uptake. Here the light is important for the photosynthesis. This but also it's light light is important for their growth, plant growth. There is light mediated growth and development is or it's give a shape morphology to the plant. That is explained by the photomorphogenesis. This why it is why we want to some area, some plants are not grown in sunlight, that are all can be grown in the shadow condition. So in that plant leaves and the growth metabolisms are depends on the not depends on the light source. So based on the plant variety and the genomics that are vary, that photomorphogenesis are vary. Okay. Then next thing is for photomorphogenesis, they need phytochromes in their cell wall, that plant leaf. That plant leaves are having the phytochromes. These phytochromes are having two types. One is phytochrome 1 and 2. In this type 1 phytochromes is a very light sensitive. If that is receiving the light source, means it will get activate, and these phytochromes are automatic inactivated by the red light. That is called as the condition called as the phytomorphogenesis. In that, this kind of activation, that light based activation, is called as the sensory effect. The plant sensing the light, due to the light sensing it will activate some pathways. So that is called as the sensory effect of the plants. In this type 1 phytochromes are mainly function in the dark condition. This is very important thing you want to know. Here they may provide us choose the best and the match the following. The phytochrome type 1 phytochrome is working in a dark condition. It's using the red light, fair light. and this example for this is a the phytochrome A. Then type 2 phytochrome, in this it's a light stable, it's not sensitive, it's light stable. The type 2 is a light stable, and it's also is function is mostly in the light condition, that means daytime. Then it's activated by the red light, normal red light. First thing is phytochrome 1 is activated by the fair red light. Here this phytochrome 2 is activated by the red light. In this for example for phytochrome 2 is phytochrome B, C, G and E. This thing is very important to know, because in this 6th unit is majorly focused on the 2 marks. This 2 marks are it's a direct, maybe the it's mostly the not problematic one, it's a theoretical one, so only I am mentioning that theoretical question. They ask the many match the following questions in the this part. So please understand once more, you will get the point. Type 1 phytochrome, it's a light sensitive, activated in dark condition, it's activated by fair red light, and example for this is a phytochrome A. Then type 2 is it's a light stable, it's activated in its function in the light condition, that is at daytime, then activated by the red light. For example phytochrome B, C, G, E. Another one more thing, extra additional point is it's in this light the transcription of phytochrome gene is inhibited, so mRNA degradation happen in the plant, so proteolytics takes place in the plant. It's a sequential process. Once type 1 phytochrome absorbs the light, that is very sensitive, right? So only the transcription of phytochrome gene is inhibited. Due to the inhibition, there was mRNA degradation happen. This mRNA degradations makes a proteolytic process. In using of the in this type 2 phytochrome, that is light stable, so that are uptake the light, and it's helpful for the growth of plant. Here they mention some notes. The phytochrome B is the most abundant light grown plant. It's present in the most abundant in light grown plants, and the phytochrome C, E are less abundant in the plants. Okay, this is the how it is response. First thing is based on the light it will grow, or it may be grown in a short form. Another thing is the types of what is the responsible for their sensor, plant sensors, that is the phytochromes, that sensible and stable for the nature. Then we will going to discuss about the response of light. This response of light, as already I said, phytochrome P is the that is the signaling molecule. In that having the two part. Here this is 1 minute. Okay. There is no structure. No. Okay. I thought that is I am included the image. That it's having two part. That is a chromophore and apoprotein. Apoproteins are it's present for their that signal transduction pathway starting induction. It's initiating the signal transduction pathway. These apoproteins are produced from this phytochromes. This phytochromes two parts, chromophore and apoprotein. The chromophores are only allowing the receiving the light. Apoproteins are initiating the transduction pathway. Here this in this image itself they mention that protein binding site, it's an ubiquitin binding site, and protein kinase. Okay, it's a overall signaling process in the plant. How it is activating, how it is look like. Okay, it's in this plant, they all many plants are morning time leaves, or it's observing the light, red light, and makes some pathway activation. At the activation, it's producing the it may produce the mRNA for a protein synthesis, and some genes are expressed, and some degradations are happen in the leaf, and the produced proteins are released in cytoplasm. This is one step in the morning. That time it's receiving the light, red light. Okay, the phytochrome is activated, but in noon afternoon time it's not activated because there is no red light. So that time that observed this LHC mRNAs are getting activated, that producing the protein for the chloroplast development. This LHC mRNA, the light harvesting compound mRNA. Yesterday class we discussed a light harvesting compound. This light harvesting compound is formation, they need a protein, right? That proteins are produced with mRNA. This mRNA, that is the LHCb protein, that produced protein helpful for the chloroplast development. Then another one is the HY5. It's a hypocotyledon elongation factor. This hypocotyl elongation is happen with this gene expression. And another thing of the this evening time, it's a morning, afternoon, and evening. And evening time it's observing the fair red. Okay, that phytochrome receiving the fair red. During that fair red absorption, that time, that producing the activating the COP protein, and it's producing the that degradation happens in the with the proteasome. The plants also having the proteasome that are makes a degradation. This degradations happen, and in this step, that observing the fair light, there is no expression of the mRNA. Here I am come back to the previous slide. Here they mentioned, right, the phytochrome C activated na, that time there is no mRNA degradation happen. That phytochrome genes are inhibited, so proteolytic happen. In this image they explain the evening time, that fair light absorbance, that is the phytochrome 1. That time that phytochromes are receiving a signal and activating the pathway that makes the proteasome, that is degradation, that mRNA degradation happen. There is no expression of LHC, that expression of TOC1 is started. Here in this morning, that observing the phytochrome 1, 2 type, that is a red light part. Then that activating the pathway, there is a no expression of mRNA happen. That mRNA expression helps to produce the chloroplast development. It helps for the plant growth. There is no expression of TOC1 gene. The main thing, the comparison, cytochrome in here, that first morning time, phytochrome 2 is activated. Due to the activation, LHCb is in active mode, and TOC1 gene is in off mode. And afternoon time, that LHC mRNA get activated, that producing the process, synthesis process are there. And evening time, it's observing fair light, that phytochrome 1 observing the fair light, so that activating the pathway, and during the activation there is no LHCb gene activation, that only TOC1 gene is activated in the process. In this step, evening, the phytochrome 1 observing fair light observing process stimulate the degradation of the mRNA. that happen with the proteasome, 26S proteasome. I think now it's clear for understanding. The next thing we are going to discuss is the structure of the phytochromes. This structure of phytochromes is differ only with this part. Only we can understand with the image. This bonding location is changed. In this phytochrome 2, that is a red light absorbance, in that they having the NH group in the opposite direction. This is the phytochrome 1, this is having the light absorbance in the that area, that upper part of the structure, phytochrome structure. Okay, this phytochrome is in trans form. That red light phytochrome 2 is that red light absorbance phytochrome, that is phytochrome 2, that having the trans form, that is called as PFR. That PFR, that phytochrome FR transform. And the fair light absorbance, it's type 1 phytochrome, it's having PR, it's a cis form. Okay, these are all things, small things, but they are that is theoretical part. In this chapter they are asking theoretical part, that only, that's why only I am mentioning this image structure and these types. Okay, role of phytochrome, that is the activation, how it is activated. Previously we discussed how it activating, how it stimulate the pathway. Now in outer that are all happen in the inner plant. In outer surface, what we are recognize, what is the role of the phytochrome? In many flowering plants used it to regulate the flowering based on the length of the day. Okay, and night, the night is the photo period. This called as this photoperiodism means some plants are producing the developing a flowering based on the length of the day, that lighting time. It's due to the length of day and night period, that plant using the flower. Okay, now for example, in morning time, and sometimes if in this duration, this month or it's our summer period, and this the early morning 5:30 itself we are getting the light from sun, so the daytime become 1 hour earlier. It's a long time. In evening itself we are, it take 7 o'clock to 6:30 7 o'clock. That's daytime is too high. But in a winter time, that in a winter time, that daytime is sun, we can see the sun after the 6:30 7. And evening itself it's disappear in the 5:30 6. So that time period is become short. Okay, based on this light, the light receiving of red light and the fair red light, that activating the plant mechanism, that called as the photoperiodism. Then and as that plant also having the circadian rhythms. Circadian rhythms, we all know, humans and plants, all everything is having the circadian rhythms. This rhythm we that having, this time is for the water uptake, this time for the sunlight uptake, this time for the production of plants. It's everything are fixed one for the plant, that fixed condition. It's their respiration. Okay, morning 6 to evening 6 we want to abduct the oxygen, then evening time that plants are uptaking the CO2 for their production, that photosynthesis production. Everything are early set in the circadian rhythms. So this circadian rhythm is designed based on the this phytochrome's activity. Then it's regulate the germinations of seeds. Okay, it's a photoplasty. Photoplasty. In this step, I want to let you know one thing. In this seed generation, you may know that seed germination happen at the mid dark condition for germination. Because that time itself that light that plants or seeds are absorbing the fairlight components, and the also it's a red light absorbance that stimulate the growth, that photoplasty, that this germination of seeds process is also called as the photoplasty. Then elongation of the seedlings and the size, shape, number of leaves, all those things are happens due to this phytochrome activity. Then strengthening of the epicot and hypocot. It's epicot hypocot's conditions are also depends on phytochrome. So here this image they mentioned, right? This hypocot elongations also happen with this happen due to the phytochromes. These are all the main role. Apart from this, once it's absorbing the light with perfect phytochrome only, that can stimulate the protein production or inhibition or degradation, that things. Okay, this one overflow, it's absorbing the light, the phytochrome is absorbing the lights, then activating the proteins, that is the PIF, that are these are the factors in the plant, and NDP, K2 and PAPS. These are all the some proteins, internal proteins present in the plant. Then it's activating the G protein, calcium, calmodulin and kinase phosphatase pathways, that gene regulations happen. This gene regulations it promotes the seed germination, leaf growth, that etiolation, photoperiod, and shade avoidance and flowering. These are all things depends on the phytochrome signaling. Another thing is next this is mode of action. How it is activated for their for our germination or their mRNA degradation. Everything is depends on the, if there is a phytochrome, there is phytochromes and 1 and 2 are there. If there is observing the fair light, that forms the X form, it's a product and it's a biological form. This according to two of them are reported, Hartman and the Bothwick Hendrick. These two research laboratories are reported. Okay, this as per their report, it's several alteration reaction pathways are happen with this phytochromes. In this image you can understand, if the light's observing the red light means for the phytochrome there is a red light in the medium, that is that phytochrome 2 is in inactive form. So due to the inactivation, that PR means cytochrome 1 producing get the product and biological production forming. And if there is a fair light means that it's in active form, that make an interaction with the product, that is ATP or NAD. That product formation helps to form the biological formation. This the two things based on the light alone. if there is a red light means that phytochrome 1 will get phytochrome 2 will get activate. Otherwise this if observing the fair light, it's in inactivated mode. And then how it is regulate the gene expression. Here you can see there is a phytochrome A. Phytochrome A means that phytochrome 1. That is here that is no direct interaction with the mRNA. This if there is a mRNA that interaction happen, that light here it's receiving the far light means that mRNA degradation happen. But here they mention the red light. The red light means that is phytochrome 2. That phytochrome 2 is make a response with the ATP, and these are all producing the degradation of the ubiquitin molecule. This autophosphorylation is happen with the phytochromes. The phytochromes are present over the gene. This serine domain having phosphate group. The phytochrome 2 observe the red light that stimulate the phosphorylation. This is the autophosphorylation in the phytochrome protein. Okay, in this step they maybe ask the question, what is the protein responsible phytochrome for the autophosphorylation? In that you want to answer, you know that concept, right? The phytochrome 2 only observe the red light, you based on the red light absorbance, that serine get phosphorylated, that autophosphorylation happen in the gene. The phytochrome gene expression means if there is a mRNA activation, the phytochrome A activation happen means that phytochrome absorbs the fair red light. That promotes the mRNA degradation. Otherwise, this phytochrome 2, there is a fair red light, that is a red light. The red light means that absorbs the absorbed by the phytochrome 2. This phytochrome 2 which promotes the seed germination, that all the process. This is another representation. What is how it is altered the gene. Due to the cardiac clock. The cardiac clock means the daytime and nighttime balancing. In the daytime it observes the red light, and that's being afternoon evening time that observing the fair red. So based on the light absorbance, it's altering the gene. Due to the transcription factors binding to the active site of the target gene, these transcription factors are promoting the seed germination, flowering, and photomorphogenesis. Photomorphogenesis means if sometimes that due to the light, that leaf size become expand. That size of the leaf is become large. That due to the phytochrome A activity, the chloroplast development and stem elongation. These are all happen due to the phytochrome's activity. Okay, photoreceptors. This is another concept in the unit 6. These photoreceptors are different type: phytochromes, cytochromes, phototropin, and UV visibles. Why I am mentioning the phytochrome alone in the previous slides now? That is very important than these other receptors. That cytochromes we discussed in the yesterday. Cryptochrome. Here this cryptochrome means it's also the another light signaling based acting compound. The phytochrome is a major activating receptors, that is a major influencing receptor in all pathways of plant. Here cryptochrome, this observing the blue and UVA light. First thing is the phytochrome. This phytochrome is observing a red and fair red. This cryptochrome, they're observing the blue and UV. Then phototropin, that observing the blue and UVA. Then UVB receptors are separated. These things are in the photoreceptor. In this, based on the light sensitive, we can tell the concept. Is another explanation. It's a red color means red based light absorbed means phytochrome. Here this is one more question. The question like they are can ask like okay, red light absorbing the photomorphogenic receptors, that are phytochromes. The blue light absorbing photomorphogenesis receptors, that photoreceptors are cryptochrome and phototropins. These two are very important. This is a pictorial representation. This phototropin is fully depends on the seed germination. Here they mention the that blue color it's depends on the seedling development and flowering. Then differential growth and light gradient. Here if you can see the phototropin is observing a blue color. This helps for the that seedling elongation. Then another is a cryptochrome. This cryptochrome is helpful for the dark condition and light conditions production. Then this phytochromes are helpful for the photoperiodism. That daytime it's look like different. It's not in observing in the daytime it's observing the red light and it's make some development, and that nighttime it's observing the fair red light, so it's not in active mode. These are all the functions of phytochromes. These are all the phytochrome A, B, C, G, E. This phytochrome A is a germination process. That this phytochrome A, B, C, D, in this all things are helpful for that seed germination. Another one is seedlings development and etiolation. This de-etiolation, it's a very light sensitive process. De-etiolation means it's producing their nodules, that roots formation, that in the development process. This and another phytochromes. This in the previous slide itself I mentioned these phytochromes are activated based on the light. This light activity, if the daytime means that absorbs the that phytochrome 2, that phytochrome 2 is in active mode. This phytochrome helpful for their production, that plant developments. Okay, this so the plant germinations happen with the phytochrome B, C, G, E, F, all those things. Okay, in this in the etiolation process means it's in a dark condition mode. Sometimes some dark conditions mode that stems are become white condition. Okay, that is weak mode, that is weak stem formation. This why it's needed for their starting stage growth. Okay, for their starting stage growth the leaves need to be strong. That stem it's not in effective mode. For their elongation, that if that we need high that a larger plant, that's a length of the plant means we need to maintain the process, the etiolation. This etiolation process, that plant producing the white color its lengthiest stem. Okay, then another thing is activation of phytochromes and ZTL F the ZTL KF1 LKP2. These things are helpful for the transition of flowering and neighbor detection and shade avoidance. Why we that this shade avoidance means that plants need light for their the light for the cell development, plant growth. You may hear the thing in your day-to-day life. Some of them tell, okay, don't place any plants nearby any tree for a tomato or small flowering plants. They won't that allow us to place that small plants near to the trees. Why? Because that shadow will avoid the avoid the lighting. It's a inhibit that stop the light from top, so there is no absorbance, light absorbance happen. So that phytochromes are not in active mode. For avoiding that, we need to avoid the shadow. And phytochrome for that photo 1 and photo 2, that UV receptors. Here cryptochrome is a CRY and 1 and 2. Cryptochromes are involved in the neighbor detection and shaped evidence. And phototropin 1 and 2, that are involved in the tropism, that is light detection process. And UV receptors also involved in the tropism. Here once again I am going to repeat. Phytochromes are involved in the germination, seed development. In this etiolation and seed development process also happen with the cryptochrome. This another thing is ZTL and FKF1 and LKP2. These two things are involved, that are along with the cryptochrome. This transition to flowering and neighbor detection, shadow avoidance are happen with these three phytochromes, this receptors. And cryptochrome 1, 2 and phototrop. The cryptochrome 1 and 2 is helpful for the neighbor detection, and phototropin 1 and 2 is helpful for the tropism. Here this is a stroma. Next topic is after this are all the signaling receptors. Okay, based on the receptor process which happen. Next thing is the stoma open. The stoma open and close response. Okay, how it is open, how it is close. In the first thing they mention the pumping means it's opening and closing. This it's releasing and intake. Pumping by H plus and ATPase increases. If there is a higher amount of AH plus ions and ATPase are there, that proton leaves the guard cells. It's released from the guard cells. That's so released. That's a n number of H plus means that will release. That is the closing. Then another step is the if there is K plus and Cl entry enter the cell along with the electrochemical gradient, invade direction. That is, invade direction means that channels are co-transporters. Here two things are there in blue light based expression. If there is a blue light striker photoreceptor, that blue light means that a phototropin 1 present in the stoma, that stoma helpful for the pumping by H plus and ATP synthase activity. That is one step is releasing of H plus ion. Another step is the allowing of K plus and Cl ions. This H plus also enter into the cell due to that blue light. Then It's a open close concept. In this open close concept, it's if there is as I mentioned earlier, if there is a H plus ions means there was the production of water, the uptake of water is high. If there is a water molecule is high, the high means in the cell there is a need of uptake. That time, that water uptaking time, the stroma cells are in open mode. Okay. If the absorbance of, there is a need of water, so it's open mode, and it's also need the will allow entering the K plus ions. Then if there is a no need of water, that is a removal of water, that is happen with the that makes the close in the stroma. That process is called as cell turgid means that is a open, flaccid means it's called as the close. This is all the overall mechanism. It's absorbing the blue light. It's absorbing the blue light, and its phototropin makes a phosphorylation process. Due to this phosphorylation process, there is kinase activity happen. It's allowed to release of H plus to membrane. So there is a hydrogen molecule transfer happen. Then another thing is here it's due to this stromal opening, this is a removal of water, right? Here mentioned it's a need hydrogen molecules are inside the cell, so it's a over strength, it's removing the H plus ions. Its stromal opening increase the torque pressure and increase the water potential, decrease the water potential. That is the waters are there, so it's need to decrease the water potential, and it's receiving the K plus ions. So there is a potential, the membrane potential is that is minus 100 millivolt. This H plus ions transfer makes the plasma membrane hyperpolarization effect. Now it's a sequential process, you may understand. Blue light receiving the received by the phototropin 1 and 2, that makes phosphorylation process. This phosphorylation process activate the kinase. This kinase it's helpful to release of the H plus ions. During this open medium, it's increased torque pressure and decrease in the water potential, and it's a recycling K plus channels. The total the potential of the stroma is minus 100 millivolt. Its stromal functions, it's a light based its function and temperature based function. In this light based function, that is there may be the red light, there may be the blue light, that based on the light that affect the factors affect the movement of the somatal means this somata that the moving, that open and close happens due to the light and temperature. The major factors affecting these means there is a number of leaves. The more number of leaves is there on the means there is a bigger surface area, that and more stomata for the gaseous exchange. It's there is a result in the greater water loss. So, okay, there n number of stomata on the leaf means there is a more water loss happen to the plant. And number of stomata it's also affected because it provides the transpiration. This it may helpful for the oxygen, that CO2 intake, and it's also affect the this water the plant growth, because the water loss makes the plant become dry. And size of leaves has bigger surface surface area is also is high, so there was the uh, fast, that the transpirations has become faster, that if there is a leaves with the surface area, smaller surface area, there is low transpiration. If there is a low transpiration happen, means there is a water water stable is high, so that plant looks become fresh. The presence of plant cuticles. The plant cuticles are, it's a waxy cuticles. This waxy cuticles are reduce the evaporations, that's overcoats. These are all the factors. Another factors, the light, temperature, wind and humidity. The humidity is depends on the water. If you maintain the water on the external surface, that will also helpful for the production. Then photoperiodism. Photoperiodism, in that they mention the few with mention with a few expectations, that the plant need a movement, that growth, reproduction, that all the things are depends on only photoperiodic. That some plants are grown in the that flowering period at morning, some plants flowering time is evening, some plants are grown in this particular month. These are all about the photoperiodism. That photoperiodism is a transducer. It's a mechanism that supplies the signals to the environment. Okay. In this photo periodism, that its phytochromes are activated due to the daytime and night. Okay, that is a major thing in the this photoperiodism. It's it's a types of photoperiodism classification. In that time, short day means that is the 6 hours. In that 6 hours, the uh, 6 hour daytime, so day length is less than 12 hours for the flowering. And long day plants, in the long day plants, we have the more than 12 hours for the flowering. Then a natural day plant means there is the is intermediate of, that is a 12 hours flowering. It depends on the flowering period, flowering time is depends on the that daytime and nights. Is another explanation about the short day and long day plants. Short day means that lighting time, that sunlight time is short, too short. Long time means that sunlighting time is high. Then biological clock, it's I'm included in this previous slides also. It's having the internal physiological system of the plant to track the environmental rhythm. It's having a 4 characters. It's endogenous. Endogenous means that innate ability of to maintain the period. The some organisms are not able to maintain the period, because humans are also the it's deviated from the clock, so it's endogenous, so it's particularly helpful for the biological function. It's also applicable human also, so you want to maintain the biological functions. It's helpful for the biological function in endogenous nature. Then another is a temperature dependent or independent. Its clock is not depends on the temperature, it depends on the light. Then biological clock is the ability to be the rest over the order to maintain the relationship with the environment. And biological clocks are the internal continuous monitor. Everything are monitored through the internal clock. Here they mention the step-by-step process. Gene expression. This gene expression makes a cytosolic calcium leaf. This photophosphorylations happen in the leaf cell, that makes the chloroplast movement. This chloroplast movement makes the stomatal opening. The stomatal opening help for the hypocotyl length. That is the length of the gene plant, and then cotyledon and leaf movement. This cotyledon leaf movement happen with the due to this step gene expression, that clock, then that petal opening in the flower, then flowering. These are all the step by step process in the rhythm, in the clock, biological clock. That system no, okay, this time happen, this flowering is happen, that is fixed system. Here for example I can say, in a jasmine, that will open their petals at this 6 o'clock, after 6 o'clock alone, after the that sunlight disappear. This is one thing. In Tamil you may know the word Pavalamalli. That plant, it's producing the their flowers at the night time alone. That is some plants also there, that it's a sleeping tree. It's their leaves are become down after the 6 p.m. These are all the biological clock function. Then uses the vast, these are the overall function. This predicts the environmental events and navigation. Okay, so it's for example in Vedantankal, you can see the animals, that birds are navigated to the Vedantankal at particular time alone. That all things are depends on the after that some period it's moved to their native place. It's all about the biological clock. Okay, it's a fixed one. Then measuring of the length of the day, that's a photoperiodism. Then timing of the reproduction. This timing of the reproduction is also one biological clock. Some plants are producing seeds at particular time alone. This all the important things we want to understand in this biological clock. The applications, they provided the application in multiple choice question and ask whether it is comes under the biological class. Understanding is very important for prediction of environmental event, navigation, orientation based position of the sun, the measurement of length of day, that timing of reproduction, and gating. Gating means timing of event occurrence and catching, that lifetime in the okay. All those things are very important in the biological class. Here having internal control and external control. Internal control means breathing, heartbeat, gut motility. Here this is all about the animal. In a plant it's a respiration, it's a photosynthesis process, all those things. In external, external means is the environment, sunlight, that water uptake, everything is comes under the then solute transport. In that solute transport, it's happen with the xylem and phloem. It's solute transport in the xylem. It's helpful for the transfer of water and dissolved ions. This the xylem sap may also contain the organic material. So only that organic material helps to transfer that water and dissolved ions. This it's depending on the different rate. It's matrix dependent and metabolic needs based one. If there is a need of metabolics in the cell, there is make a solute transport. Okay, it's a phloem based one. The first one is a xylem based one. The xylem based one is it's helpful water and dissolved ions. It's transport to the transpiration happen through the xylem. It's xylem sap. It's also contains organic material with a low concentration, and its substance to move different rate. It's depending on the matrix development and metabolic needs. Then it's based on the phloem. The sugar must loaded and to this tube, that zinc tube. It's that sugars are loaded on the tube, and depending on the species, that's sugar may move that symplastic and both symplastic and apoplastic pathway. This symplastic and apoplastic pathway we can transfer the ions, that is molecules. This modifications enhance the solute movement between the apoplast and symplast. Here this condition for the movement, there is a leaf, there is a nutrient level in the its reduction of nutrients level in the shoot that makes the activation of intake, that is the intake make the phloem, make through the phloem, that nutrition uptake in the root, so that the low intake happen in the root, so nutrient level in the soil is low. For activating this, we need to improve the nutrients level in the soil, so uptake is high, the phloem become activated, the nutrition level in the shoot become high. This is the nutrient translocation in the shoot. Then mRNA translocation in the root, the nutrition translocation in the shoot, that mRNA translocation in the root. How it has happened? That in that pathway, the target gene level is want to be maintained in the root. After that, the mRNA translocation has become high, the mRNA synthesis is also high. Here the target gene level is low in the root. That time that mRNA translocation happen to root, the synthesis will become high. These are all the step-by-step process. So in the phloem based pathway, we can produce mRNA, miRNA, and miRNA it's translocated to the root. The miRNA is a microRNA, not a mRNA. A microRNA. This microRNA helps to produce the target gene. Produced from the target gene, it's helpful for the production of proteins and amino acids, metabolites. This is a translocation process. The source is the cells, then phloem. That phloem it's based on the zinc, that is the root cells. Here step by step process. Here you can see the image cell surface. So this is the small veins of plants. Through this plant vein, it's observing the mesocells and plasmodesmata. These are all the in between the plant connecting area. In that area too, they are observing the molecules and transfer to another cell, that is a movement. Okay, that source, this source is helpful for the this is a apoplast condition. This apoplast condition, here the higher concentration of H plus ions. In this layer, the lower concentration of H plus ions. So the intake of H plus ions make a sugar symphosters, that sugar molecules are it's make a ATP, that in that step also the ATP conversion also happen in the cell. This ATP conversion make a release of H plus ions. That sugar molecule intake is helpful for the H plus ion intake, and that release of the ATP to ATP conversion is makes a H plus ions release from the low concentration to high concentration. Okay, these are all these things in the apoplast function. Apoplast function is based on the hydrogen ions exchange and sugar molecule exchange. So water uptake, how it is water is uptake into the cells. How the cells are uptake, the water is uptake through the osmosis process. In the xylem, the transpiration systems are activated. This in the phloem, its water molecules are translocated through the leaf cell to the leaf cells and zinc cells. From the leaf to its zinc cell. Here the loading of sources from the loading of sources is sugar. Here they mention the sugar, that is a sucrose molecule. That sucrose molecule is source from that leaf cells. That leaf cells are act like a tube and increase the solute concentration and decrease the water concentration for the osmosis process. here the concentration is become the exchange happen with the osmosis process. That water uptake also happen by the osmosis process. It happen with the it's normally it's flow, it's normal in the xylem but abnormal in the phloem. Then it's for example, here it's very well exampled one. Here you can see the molecules, small molecules that may, here they mention nanoparticles, that may be the water, that may be the ions, everything. If that is added to the irrigation land, that plants are absorbing through the that mycorrhiza and organic matters or solutes are uptaken through the plant root. Okay. Here you can see the all the components in the root, and here the organic matters and salt ions from this nanoparticle, that is a molecule, soil molecules are uptake through the root. Another thing is spraying. Spraying means that spray is that ions are water, so only they are spread the water through over the plant. That leaves also absorb the water. The two way of the translocation, one through is a root, another way is a leaf. Here another thing is the through the plant. Here they having the sequential plasma membrane on the cells of that particular plant cell. In that plasma membrane allows the some molecules by endocytosis or pores formation or receptor based interaction or plasmodesmata process. These are all the sequential processes. These are the type of translocation. One is translocation happen with the root, another pathway is a leaf pathway, and another one is the plant movement. Okay, this plant movement is a aplastic pathway and a symplastic pathway. Here here in this aplastic pathway means that plants are the dotted lines are called as the symplastic, and the straight lines are the aplastic. Here aplastic is the, here one molecule enter in this region, it's move through the cortex to the endometric layer. In this endometric layer, it's passed through the endometric layer and move to the region called as this phylum. Then after reaching the phylum, that phylum region, it follows the symbiotic pathway. And symbiotic pathway using this companion cells that moves to the xylem. In that xylem, that again makes the aplastic pathway. Here you can understand which cells are allowing the apoplastic pathway. Apoplastic pathway, they are using the epidermis, cortex, and endodermis, and the endodermis to the phloem. After the reaching the phloem, that follows the symplastic. In that symplastic process, that allow the molecule to enter into the xylem. In the xylem, that makes the apoplastic process. Here some molecule can enter directly through all the membranes and reach the xylem. That is one thing. Then penetration having endocytosis may be happen with the ions, and pore formation, and there is a receptor based one, and plasmodesmata process. Plasmodesmata means make a connection between the external surface to internal membrane. And photoassimilators assimilates. This assimilates is molecules which are helpful to transfer the signals to sheath cells to that Schiff elements. Here the bundle shells or sheath cells are there that produce the release the sugar molecule. That sugar molecules are transfer to the cells. During the transfer, that hydrogen plus ions also transfer. This process called as the apoplast. In that apoplast, after the apoplast, that sugars are moved to the Schiff elements. The Schiff elements are the another cells transport. Then amino acids, if they are receiving the amino acid from the bundle sheathal cells, that amino acids are also allowed entering of hydrogen plus ions. These hydrogen plus ions are used to and released in the multiple count, because during this this amino acid interaction, the hydrogen plus ions are utilized by the mitochondria for the production of the various compounds. This ATP to ADP function helps to produce more number of hydrogen plus ions. So only they mention the n number of hydrogens are moved. Then the phytochemicals production. In this phytochemical production, we have different type. This is a phenolic production, biosynthesis of phenol in the plant. How it is produced? As I mentioned in the previous class, that plants also producing the amino acids. That amino acids are helpful for their phytochemical secondary metabolites production. This secondary metabolites, phenylalanine helpful, it's converted into tranexamic acid. Then it's converted to p-coumaric acid, that it converted into the coumaryl CoA synthase. After this synthase pathway, it's produce the phenolic compounds, and during this pathway, it's using the cinnamic acid helps to produce the benzoic acid derivatives, and coumaric acid is helpful for the caustic acid and other simple polyphenols production, and coumarin production, and lignin precursors production. These are all the sequential process. And this is another way. Here the erythrose phosphate and phosphoenol pyruvate, these two acids are involved in the succinic pathway. It's helpful for the production of phenylalanine. It's a one step previous to the phenylalanine production. It's also happened with the in a plant. That erythrose 4-phosphate and phosphoenol pyruvate acids that are undergone in the succinic acid pathway, it helps to produce phenylalanine. This phenylalanine helpful for the production of the lignin, condensed tannic acid, flavonoids, miscellaneous phenols, that is means unknown phenols. That acetyl CoA, that the end product is a CoA, right? This CoA is acetyl CoA is helpful for the using the malic pathway and producing phenols. So using this polyphenol phenylalanine, we can produce phenols, flavonoids, tannins, lignins. This is the thing we want to understand. Using acetyl CoA, the malic pathway, through the malic pathway, it's producing the phenol. Another way is tannic, using the succinic pathway, that pathway produces the gallic acid, it's produced the hydrolysable and tannins. Here terpenoids production. In this terpenoid production, we can use the that enzymes called the acetyl CoA, that HMG CoA, it's produced the mevalonic acid. Then it's produced the isophenol phenyl diphosphate. This isophenol diphosphate is helpful for the production of the isopropane. These all things are helpful for the production of terpenes. This is the terpene compound. This is helpful for the it is also happen it's produced from the pyruvate pathway. Pyruvate and glyceraldehyde 3-phosphate, this forms the deoxyxylose 5-phosphate, methyl erythrotiol phosphate, and it's producing the dimethyl diphosphate. These two compounds are forms the terpenoids. This pathway involves malvalonate pathway and methyl erythritol pathway, phosphate pathway. These two pathways are only terpenoids production. Here another way is using of H2O2, that photosynthesis are happen in the plant. The photosynthesis are helps to produce carbohydrate. This carbohydrates are helps to produce polysaccharides, nucleic acids, pentose path. Activating the succinic acid pathway. That carbohydrates are undergone the hydrolysis glycolysis pathway, producing the phosphoenol pyruvate that produce the pyruvic acid. Then acetyl CoA production, malvalonic acid production. This malvalonic acid helpful for the terpenoid, steroid, carotenoid production. Okay, that from using this carbohydrate, that erythrose 4 phosphate become activated. Using this erythrose 4 phosphate pathway, that lignin, coumarin, phenol, flavonoids are that produced. It's a one way. Another way, use this succinic acid is it's also helpful for the production of aromatic amino acids, that is called as the tyrosine, phenylalanine, and tryptophans. This produced tyrosines are helpful to produce alkaloid, proteins, and enzymes. And one more thing, using the pyruvic acid and acetyl CoA that are producing the alpha amino acids, aliphatic amino acids. These aliphatic amino acids are using alanine, valine. These aliphatic amino acids are using alanine, valine. This produce the alkaloids, proteins, enzyme. These aromatic amino acids also there, tryptophan, tyrosine, and phenylalanine. That is also helpful for the production of alkaloid, proteins, and enzymes. Then another thing, using this acetyl CoA that producing polyketide and fatty acid that produce helpful production of polyphenol and prostaglandins in the plant. These are the overall production image. It's a very important thing. Please refer after the PPT receiving from the mail or Google Classroom. Then stress are there. Many stresses are there. In that stress itself, that makes plants to produce some phytochemicals or stop the production of phytochemicals. That is secondary metabolites. This how would the stress are happen? It may happen with the heat, chemical, salinity, or coldness, and these all the things are make a secondary stress. These are first primary stresses are chemicals, pollution, salinity, heat, cold, droughts. These are all make a secondary stress called as the osmotic stress and oxidative stress. This osmotic stress and oxidative stress are it's disturbing the osmotic stress means that is the intake of water that will also affect. Then it's damage the cells and its change the structure of production and protein and membrane. Then another thing is oxidative stress. Is the osmosensors. This osmosensors are getting affected, so that production that make a unbalanced intake of calcium ROS, reactive oxygen species. All the things are happening. Then transcription factors also affected. The transcription control. The transcription factors means that is a mRNA production, the cytochrome activity, everything were distracted. Then stress responsive mechanisms are involved with detoxification happens, and osmoproteo protection. This osmoprotection is affected. Then chaperone function will get affected, and water and ion movement is also will maybe affected. These are all the possible way of abiotic stress. Abiotic means it's a apart from the biological function matters. Here this is a simple form. The signal transduction is affected. The transcription control by transcription factor, so it sequentially affect. Then so gene expression will get affect. That gene expression is modify the detoxification, osmoprotection, chapter on function, and water ion movement. This gene expression function helps to modify that make a change in the stress response, protect cellular structure, and restore the uh, uh, hemos hemostasis process. This helps to tolerance. Tolerance means make a tolerance, balancing. This gene expression, after the gene expression, that makes a tolerance. Very important thing, that this till this step we are discussing the what is the possible way of this stress. So that steps makes a change in the this four process. That four process helps to balance the plant genetic structure and function. That structure is called as a tolerance. Here, okay, it's maybe the biotic or abiotic stress, but that stress is that makes the uh, mutation in the ABA pathway, phytochrome interaction, plant activation, and programmed cell death become maybe affected. Cell small RNAs are affected. Infection of biology and image that may plant's leaf may get infected. Then another thing is here in this side, these are all the abiotic conditions. So alter the gene expression, impact of other hormones or influence, it's very exhaustion. Then another is the abiotic conditions makes the osmotic disruption and AB receptor. Here itself they mention that oxidative stress, make the oxidative stress and osmotic stress. This is the biological function stress. This is in the biological stress, it's make it's affect the signal transduction. So that directly affect the gene expression, or it may be the so we want to produce the anti-infection. This anti-infection, it's a sequential process. You can this anti-infection, how it is produced. Using this strings, okay, once it is enter means transcription, that signals are the CDPK and H1 ATPase and CRT, that are pass a signal to gene expression. That gene expression make energy, that activating the some enzymes that activate the due to the activation, metabolisms or protein metabolisms are stimulated, and protein synthesis and degradations are allowed. That during this is ribosomal proteins are activated, the translation elongation happen with the TU factor, and ubiquitin like proteins are activated. This activator helps to produce the anti-infectant. That anti-infectant act against to the that biological molecule. This is one thing. Another one thing is the plant defense systems are activated. Plant defense system is a PBZ1 is a probenzol molecule. It's MOXOOSM viruses. These are all against to this. These are all the foreign particle. The probenzosol, MOXOOSM, and virus particle. These are against to that. And is OSPR10. It's a again. This is the kind of the cytosine. It's a polymer. Then OS, these are all the things are chitinase, it's all present, it's all chitinase all present in the over the fungal surface, that these are all the chitin present in the cell surface of the fungus. So this chitinase helps to degrade the fungal infection. These all the things are happens against to the biotic stress. Then reactive oxygen species also produced. This SOD and APX, this catalyze. These are all the reactive oxygen species. This helps to avoid the biotic stress. This, for example, we can tell this plants are producing the some anti insect property, because that plants are producing the resistance against the that insect. This is all about the unit 6. Unit 6 over much topics, because it's a new one. You want to understand well. The 11th unit and 8th units are remaining in the session 2. We have 2 days in the session 2, that is the 22 and day 22 and 23 is the remaining days in the session 2. Tomorrow I'll try to cover the unit 11 and unit 12. This very important in the that unit 6 is very important in the session 2. So it's overall I'm covered many things. I'll send some notes today also. Please refer that for your understanding. And another thing is NCERT 11th and 12th book will having all the informations. Thank you for your cooperation and patience. Today I'm taking much time because that starting later only I'm started some problem in the software. Thank you for your patience. If you have any doubts, please ask in the chat box. Yeah, Cryptochrome. It's Cryptochrome.